Hello everyone, welcome back to my YouTube channel Science Academy, where science made simple and easier. In today's biology session, we are going to complete 20 most important and repeated MCQs on topic enzymes. These MCQs are very important for biology IGCSE grade 9th and 10th level. Let's get started. Question 1 is, what is an enzyme? The options are a type of carbohydrates, B, a biological catalyst made of protein, C, a lipid that speeds up reaction, D, a nucleic acid. And the correct option is option B. Enzyme is a biological catalyst made of proteins. Exam tip: enzymes are proteins that act as biological catalysts. They speed up reactions without being used up. Question 2. What is the active site of an enzyme? Option A, the part that gets destroyed after a reaction. Option B, the region where the substrate binds. Option C, the area that produces ATP. Option D, the outer membrane of the enzyme. And the correct option is option B, the region where the substrate binds. Active site is the region where the substrate binds. Exam tip: the active site has a specific shape complementary to the substrate. Key to the log and key model. Question 3. Which model explains enzyme substrate specificity? Option A, fluid mosaic model. Option B, double helix model. Option C, lock and key model. Option D, cell theory model. And the correct option is option C, lock and key model. Exam tip: lock and key enzymes. Lock has an active site that only fits its specific substrate key. Question 4. What happens to an enzyme at a very high temperature? Option A, it works faster permanently. Option B, it becomes denatured. Option C, it becomes a carbohydrate. Option D, it multiplies. And the correct option is option B, it becomes denatured. Exam tip: high heat breaks hydrogen bonds holding the enzyme's 3D shape. The active site changes shape is equal to denatured. Question 5. What does denaturation means for an enzyme? Option A, the enzyme speed up. Option B, the enzyme is destroyed by acid. Option C, the shape of the active site is permanently changed. Option D, the enzyme becomes more specific. And the correct option is option C, the shape of active site is permanently changed. Exam tip: denaturation is irreversible change in enzyme shape. The substrate can no longer fit the active site. Question 8. At what pH do most human digestive enzymes work best? Option A, pH 2. Option B, pH 7 neutral. Option C, pH 9. Option D, pH 14. And the correct option is option B, pH 7 neutral. Human digestive enzymes work best at pH 7 neutral. Exam tips: most enzymes have an optimum pH of 7. Exception: pepsin works best at pH 2. Stomach acid. Question 7. How does increasing substrate concentration affect enzyme activity up to a point? Option A, decreases activity. Option B, has no effect. Option C, increases the rate of reaction. Option D, denatures the enzyme. And the correct option is option C, increases the rate of reaction. Exam tip: most substrate is equal to most enzyme is equal to substrate collagens. Faster rate until all active sites are occupied. Saturation. Question 8. What is the role of enzymes in digestion? Option A, to absorb nutrients. Option B, to break down large food molecules into smaller ones. Option C, to produce bile. Option D, to transport oxygen. And the correct option is option B, to break down large food molecules into smaller ones. Exam tips: digestive enzymes break down large insoluble molecules, for example starch to glucose, for absorption. Question 9. Amylase break down which molecules? Option A, protein to amino acids. Option B, fat to fatty acid and glycerol. Option C, starch to maltose or glucose. Option D, DNA to nucleotides. And the correct option is amylase breakdown. Which molecules? Amylase breakdowns starch to maltose glucose. Exam tips: amylase is produced in the salivary glands and pancreas. Starch to maltose, then glucose. Question 10. Which enzyme digests protein? Option A, amylase. Option B, lipase. Option C, protease. Option D, maltase. And the correct option is option C. In which enzymes digest protein? So protease enzyme digests protein. Exam tips: protease breaks protein into amino acids. Examples: pepsin to stomach, trypsin in small intestine. Question 11. Lipase breakdown fades into which product? Option A, glucose and fructose. Option B, amino acids and water. Option C, glycerol and fatty acids. Option D, starch and maltose. So the correct option is option C, glycerol and fatty acids. Exam tip: lipase, lipids, fats, oil, glycerol plus fatty acids. These are absorbed into the lymphatic system. Question 12. Where is pepsin active and where does it digest? Option A, small intestine or starch. Option B, stomach proteins. Option C, mouth fats. Option D, large intestine or cellulose. And the correct option is option B, it's stomach to proteins, stomachs and protein. Exam tip: pepsin is a protease that work in the acidic stomach pH 2 to break down proteins. Question 13. Why do enzymes stop working below their optimum pH? Option A, they run out of substrate. Option B, ions alter the shape of the active site. Option C, temperature becomes too high. Option D, they are absorbed by cells. And the correct option is option B, ions alter the shape of the active site. Exam tips: extreme pH high or low disrupts ionic bonding and hydrogen bonds, changing the active site shape. Question 14. An enzyme's rate of reaction doubles as temperature rises from 10 degrees centigrade to 20 degrees centigrade. This is because option A, more enzymes are produced. Option B, molecules move faster and collide more often. Option C, the active site enlarges. Option D, substrate is converted to enzymes. And the correct option is option B, molecules move faster and collide more often. Exam tip: higher temperature is equal to greater kinetic energy to more frequent enzyme substrate collisions per second. Question 15. Which statement about enzyme concentration is correct? Option A, increasing enzyme concentration always increase rate indefinitely. Option B, rate increases until substrate becomes the limiting factor. Option C, more enzymes always denature faster. Option D, enzyme concentration does not affect rate. So the correct option is option B, rate increases until substrate becomes the limiting factor. Exam tip: more enzymes speed up reaction until substrate runs out. The rate relative substrate limited. Question 16. What is meant by the induced fit model of enzyme action? Option A, the enzyme is destroyed after use. Option B, the active site changes shape slightly to fit the substrate. Option C, the substrate is forced into the active site. Option D, enzymes work best at low temperature. And the correct option is option B, the active site changes shape slightly to fit the substrate. Exam tip: induced fit refined model, active site molds itself around the substrate for a better fit. Question 17. Catalase is an enzyme that breaks down hydrogen peroxide. What are the products? Option A, water and carbon dioxide. Option B, water and oxygen. Option C, hydrogen and oxygen. Option D, glucose and water. And the correct option is option B, water and oxygen. Exam tips: hydrogen peroxides break into water and water plus oxygen. Catalase is found in liver, potato and most living cells. Classic practical. Question 18. Which factor does not directly affect enzyme activity? Option A, temperature. Option B, pH. Option C, substrate concentration. Option D, the color of the solution. And the correct option is option D, color of the solution. Exam tip: only temperature, pH, enzyme and substrate concentration after enzyme activity. Color is irrelevant. Question 19. What happens to enzyme activity when all active sites are occupied? Option A, active activity continues to increase. Option B, the enzyme denatures. Option C, the reaction rate reaches its maximum. Vmax. D, more active sites are produced. Correct option is option C, the reaction rate reaches its maximum. Vmax. Exam tip: at saturation Vmax, adding more substrate has no effect. Every active site is already busy. Question 20. Salivary amylase stops working when food reaches the stomach. Why? Option A, there is no starch left. Option B, the acidic pH denatures salivary amylase. Option C, the temperature is too low. Option D, water breaks down the enzyme. And the correct option is option B, the acidic pH denatures salivary amylase. Stomach pH 2 is far from amylase optimum temperature, that is 7. Denaturing its pancreatic amylase continues in the small intestine. At last, here are the key enzyme concepts to remember. These 20 MCQs are the most important and most repeated MCQs for any kind of exam, for IGCSE, for grade 9, for grade 10th level. So here are the some key concepts. First, that is the definition of enzymes. Enzymes are biological catalysts made of proteins. They speed up reactions without being used up. Then temperature rate increases up to optimum 37 degrees centigrade in human, then drops sharply as enzyme denatures. Then digestive enzymes: amylase to starch, protease in proteins, lipase to fats produced by salivary gland, stomach, pancreas. Lock and key: substrate fits specifically into the active site. Shape is complementary is equal to high specific pH. Each enzyme has an optimum pH extremes. Denature it: pepsin pH 2, amylase pH 7. Rate and saturation: more substrate enzymes is equal to faster rate until saturation point Vmax is reached. On the topic enzymes, a detailed video has been already uploaded in my channel. You can watch from there and you can clear your concepts on enzyme. Thank you so much. Stay tuned for more videos. Stay curious, keep learning, and never stop exploring. Don't forget to subscribe, like, and share.